Hello, and thank you for attending today's webinar, where we'll be focusing on both fluorescent and chemiluminescent Western blots. Today's discussion will include the benefits of both fluorescence and chemi, some exciting new dye and probe technologies, as well as a unique, versatile, and cost-efficient solution for image documentation and analysis. My name is Mike Capps, Marketing Product Manager with UVP, and I'll be your webinar moderator. UVP is a Southern California-based bioimaging company which manufactures a variety of life science and imaging-based products and is also the sponsor of today's webinar. Before we start, let's take a moment to ensure that everyone is familiar with the webinar control panel, which you'll see on the right side of your screen. You can minimize this panel by clicking on the right-facing red arrow near the upper left corner on the panel. You can then expand the panel by clicking the same red arrow. In addition, we encourage you to submit questions using the question pane located near the bottom of the control panel. Simply type your question in the chat box and click the send button. We'll be saving questions for our Q&A period at the end of the webinar, and for all questions that we aren't able to answer today, we'll be glad to reach out to you on an individual basis following the meeting. So with that, let's get started. Our first presenter today is Dr. Sean Gallagher, Vice President and Chief Technology Officer at UVP. Dr. Gallagher has 26 years of experience in biotechnology with an emphasis on instrumentation and applications development. Currently, he is on the editorial board for the laboratory manual Current Protocols in Essential Laboratory Techniques, part of the Current Protocol series from Wiley Current Protocols. Welcome, Dr. Gallagher, and thank you for joining us today.
Well, thank you, Mike. It's a pleasure to be here. For today's presentation, I'll be covering a range of overview topics specifically around the visualization and applications of protein or Western blots. In addition, I'll spend some time comparing both CCD and film-based imaging techniques for Western blotting using chemiluminescence. Researchers still use film due to historical reasons. Digital CCD imaging is more sensitive and gives a researcher a wider range of accurate data, and that will also be covered in detail. Dr. Roberts will be talking in detail also about fluorescent blotting in the NIR later in the presentation. Lastly, we will have a discussion on the feature sets for CCD-based imagers that are best suited for chemiluminescent and NIR protein blotting. The type of visualization method used to develop a protein blot is usually also used to identify the type of technique used for protein blotting. So, for example, the techniques shown here are chromogenic, chemiluminescent, and fluorescent. So for chromogenic blots, two enzymes are used. This is also true for the chemiluminescence, and they are horseradish peroxidase and alkaline phosphatase. For the HRP shown here and AP shown here. Chromogenic development is still popular and straightforward method for developing a protein blot. Because it is simple, deposits a colored reaction product on the membrane and can be visually inspected, producing a dark brown, dark purple, or blue-gray stain depending on the enzyme used. However, the trade-off between low cost and straightforward techniques such as chromogenic development is that they are not particularly sensitive and they do not have a wide dynamic range. Some of the more popular reagents for this include diaminobenzene, nickel chloride, and TMB for the horseradish peroxidase, and BCIP, NBT for the alkaline phosphatase techniques. Now, to improve the sensitivity and linearity, and in fact to create a better way to store data, many labs have moved to chemiluminescent imaging. With CCD capture in particular, you get a wide dynamic range and great deal of sensitivity using the same two enzymes, HRP and alkaline phosphatase. Now for HRP, it's typically a luminol-based technique, and once the blot has been developed with the horseradish peroxidase tag, the luminol reagent is added, and a blue light is produced that can be captured by a CCD digital imager. As I mentioned, the film is also used, but film has a number of limitations, which I'll outline later in the talk. Now, alkaline phosphatase techniques using dioxazine phosphates for chemiluminescence deserve special mention because they tend to have very prolonged and stable light emissions, and in some cases lasting a day or longer, making them very easy to image and to repeat the imaging in order to get the ideal dynamic range for the blot. Now, fluorescence blotting, particularly in the NIR, will be covered by Dr. Roberts in much detail later on in the presentation. Quick comment about fluorescent techniques is that they are really very straightforward. There's no development step. You simply add your antibody that has a tag and visualize the fluorescence. You do need a excitation and emission setup for exciting the fluorescence and detecting the correct fluorescence. But the advantage of fluorescence is that you can do multiplexing, detect more than one sample at a time, more than one protein at a time on a blot, and they're extremely stable. In the dark, they can last for many months and be reimaged at any time. The Western blotting applications cover a range of protein science. They start with subcellular localization of proteins. Where is a protein? What is it doing? That can be identified via immunoblotting. Protein purification. Is a protein being purified during the various steps of my process? Are there degradation products? Of course, antibodies are used throughout diagnostics for identifying proteins that are either markers of disease or actually resulting in the disease themselves. A good example of that is HIV testing. And mentioned gene expression earlier, but all the full range of tracking, not just the question is not whether a gene is turned on or off always, but does it actually produce a protein? And so antibodies can tell you the amount of protein is produced and whether it's being degraded quickly in the cell. Post-translational modifications are also easily identified with Western blotting applications, and this includes the very important area of intracellular signaling molecules, typically done through internal phosphorylation, protein phosphorylation in the cell. And so antibodies are available that are specific for the phosphorylation sites of the protein, so you can actually determine whether a protein is phosphorylated or not with the range of available antibodies. Now, Western blot workflow these days is really very straightforward. It starts with a SDS page or SDS polyacrylamide gel electrophoresis, separation of the proteins. This could be a lysate, a sample you're interested in that has a specific protein you're trying to identify. Once the gel is performed and the proteins are separated, those proteins are transferred onto a blot membrane. And this blot membrane is typically nitrocellulose or PVDF, and it's a porous substrate which binds proteins. And it makes the reagents available to bind to the separated proteins. See, once during electrophoresis the proteins get separated, but there's no way for the antibodies or visualization reagents to make their way into the gel. They can't diffuse in readily. Once the proteins have been transferred onto this nitrocellulose and PVDF membrane, it makes it very easy to apply reagents, wash them, and identify the protein without any restrictions from the gel. But once the blot is done, then the next step is to actually block all the remaining unbound or the sites that don't have sites on the membrane that don't have protein present. And that is typically done with a blocking buffer containing a non-specific protein. Sometimes it's fish gelatin, sometimes it's casein, milk protein. There are a range of proprietary blocking reagents available as well that give very clean backgrounds. Once that process has happened, then you can add your primary antibody, which is specific for the protein of interest. That then binds to the separated protein on the blot. and wash that away. Apply your secondary antibody, which is a more general antibody specific for the species that the primary was developed in. So if the primary is a rabbit antibody against your protein, the secondary could be a goat anti-rabbit. And the secondary antibody contains the tag of interest, the tag to visualize the sample, either a fluorescent tag or an enzyme. After the secondary has been added, wash step is done, and you can go right to detection. And I'm going to discuss the chemiluminescent detection right here. It's really a simple matter of adding the chemiluminescent substrate. If you're working with luminol or HRP, you want to be ready to immediately image the blot because the signal can drop off very quickly. And with CCD imagers, digital imagers, it's very easy to get a quick preview, see that you've got a signal, and go ahead and take the image to get your quantitative Western blot with chemiluminescent detection. Size markers are available that also bind antibodies and make it very convenient to see the molecular weight. Alternatively, you have to mark the size markers independently. Now, you can also expose this blot to film, and as I'll cover a little bit later, I'll explain why film is really a very insensitive and technique that has a limited dynamic range. So in summary, for chemiluminescent blotting, you can see that I have run a sample and I have a protein on the membrane shown right here, and I can come in this case it's alpha tubulin. I can come in with a primary antibody. In this case it's a mouse directed, developed specifically against tubulin, mouse anti-tubulin. Then to identify this mouse antibody, I come in with a goat anti-mouse, which contains the enzyme horseradish peroxidase. I add substrate, produce light, and then that can be detected over here with a CCD digital imaging system. Now, historically, film-based chemiluminescent detection is really the original way to do this because there really weren't decent digital imaging systems available. But it became apparent very quickly to anybody working with film that it's a very difficult technique to use because simply moving the membrane around on the film can give you a contact exposure error. So just as you were positioning the membrane, or as I positioned the membrane on this film, you could see that I got a replicate image. And you also have to have multiple processing steps in wet development in a darkroom. Lastly, you have to still scan the film in order to do some quantitation. Now with CCD-based chemiluminescent detection, the key thing here is that CCDs provide not just equivalent but improved sensitivity. It can be quite dramatic. And CCDs also provide a much wider dynamic range, so you can detect a brighter signal and a dimmer signal accurately with CCDs, and you cannot do that with film. It has improved linearity, and you have much shorter exposure times with CCDs because of their sensitivity. Now, in order to effectively do CCD-based chemiluminescent detection, we use cooled scientific cameras and lenses which are low f-stop lens, very fast, they let through a lot of light, and we are using modern CCD. In this case, we typically use a 4 to 8 megapixel CCD that is cooled. Now just to illustrate how important it is to be able to use CCD digital imaging techniques as opposed to film, I want to show an example of this using a standard light source. This is a gaseous tritium light source with 5 different apertures. Each one of these apertures contains a different neutral density film, a neutral density filter, and then giving you a range from bright right here down to dim, with a little over 600-fold change. The gaseous tritium light sources are very, very stable, so you can use them over days and weeks and months and get very accurate data. And we use the same standard with a CCD imager and also with film. And you can see that with a 60-second exposure with the lowest sensitivity, 1-by-1 binning on the digital imager, we can see all 5 apertures, whereas film you can only see 3, and you lose the 2 faint ones. With film, if you even go to 300 seconds instead of 60, you still don't see these last 2 apertures, and you rapidly saturate this. This can all be quantitated, and if you take a look at aperture 3 and do background subtraction and analyze this via area density analysis, you can see that with the CCD at the low sensitivity 1-by-1 setting of binning, get 2600 grayscale counts brightness, and film shows about 2800. So at its lowest sensitivity, CCDs are as sensitive can give you the same levels as film at this middle number 3 aperture on the standard. However, if I increase the sensitivity of the CCD by using a technique called binning, I can easily dramatically increase the amount of detection I have, and it far surpasses that of film, even when the film is run 5 times as long. And on top of that, CCDs are more sensitive, so you pick up the faint signal, whereas on film it's almost impossible to get down to this number 5 aperture, the faintest signal. Now, to show this in a little less abstract way, this is a real Western blot, and you compare the 1-by-1 binning on CCD at 30 seconds with the more sensitive setting of 4-by-4 binning, 30 seconds, to that of film at 30 seconds. And you can see that the 4-by-4 binning gives you more sensitivity on this dilution series of proteins blotted and identified with chemiluminescent protein blotting techniques. CCDs also have a much wider range of linearity, which is another way of saying they see both bright and dim portions of the sample without saturating. And you compare the range of linearity over a wide range of concentration with CCD to film, and you can see the film under the same conditions saturates out very quickly to give you much less linear region and much less dynamic range.
Well, thank you very much, Dr. Gallagher, for this brief introduction to Western blotting and some background on CCD versus traditional film-based imaging. And thank you as well to all of our audience members who've been participating by submitting questions for our Q&A period, which we'll be getting to shortly. At this point, I'd like to introduce our second guest, Dr. Lori Roberts, who's the principal scientist in the bioscience group at BioTeam Inc. Dr. Roberts completed her PhD in biomedical sciences at the University of California, San Francisco, and she has 12 years of experience in cell biology research in the biopharmaceutical industry and joined BioTeam in 2010, where she supervises biological testing of BioTeam's new fluorescent reagents and technologies. Thank you for joining us, Dr. Roberts, and I'd like to pass control over to you now for a deeper look into the technology behind the science of fluorescent Western blotting, as well as for in-depth information regarding BioTeam's next-generation CF fluorescent dyes. Dr. Roberts?
Thank you, Mike. I'd like to start by just going over some of the advantages of fluorescence detection for Western blotting compared to chemiluminescence. The main advantage of using fluorescent dyes is the ability to perform multiplex detection. That is, to detect more than one target on the same blot simultaneously using dyes of different colors. Because chemiluminescence produces only one color, each target must be detected independently, and the blot has to be stripped of the antibodies and reprobed in order to detect a second target on the same blot. So you may be familiar with fluorescent dyes for other applications like fluorescence microscopy or flow cytometry. These dyes really haven't been used in Western blotting primarily because they have a lower sensitivity compared to chemiluminescence. They have been shown to have a broader dynamic range, and they have the advantage of a stable fluorescent signal. But due to the lower sensitivity, they really haven't caught on in terms of Western blotting. In contrast, in recent years, a new class of dyes called near-infrared fluorescent dyes, which are dyes with very long wavelength emission, have been applied in Western blotting. The main advantage of these dyes is that biological samples have very low autofluorescence in the near-infrared region, and this allows for highly sensitive detection and quantitation. So with near-infrared fluorescent dyes, Western blotting detection actually has a higher sensitivity and a broader linear range compared to chemiluminescence. These dyes come in different colors, allowing for multiplex detection. The one downside of these is that you do need to have a specialized imaging system that has the proper excitation and emission filters for imaging these dyes. So this is just an example of dual color multiplex imaging. So in this example here, a cell lysate containing is blotted to a membrane. And here it's showing two protein targets now present on the membrane being detected simultaneously with antibodies that are raised in different species. So for example, this protein COX-4 is being detected with an antibody raised in rabbit, whereas tubulin is being detected with an antibody raised in mouse. And then secondary antibodies against each species that are conjugated to different colors of near-infrared fluorescent dyes are then used for the detection. And so here, this is a dye called CF680 detecting COX-4, and simultaneously another dye, CF770, detecting antitubulin. The Western blot workflow for the first stages of Western blotting is identical for both chemiluminescence and fluorescence detection. However, fluorescence does offer some advantages in terms of simplifying workflow. And so basically, once the blot has been incubated with the primary and secondary antibodies, whereas with chemiluminescence detection, substrate has to be applied to the membrane and then exposed to film or imaged on the CCD camera within a fairly limited period of time after applying the substrate. With fluorescence detection, the blot is simply dried, and the fluorescent signal is very, very stable. So the blot can be imaged within days, weeks, months, or in our experience here, actually years after performing the blotting. Because it's multiplex detection, you have a one-step detection of your targets and internal controls, or to two different targets. There are fluorescent markers available, so fluorescent markers can be imaged simultaneously. Again, eliminating the need to strip the antibodies from a blot and reprobe, which you have to do if you want to do multiplex detection, multiple target detection with chemiluminescence. So now I'd like to introduce Biodium CF dyes, in particular for near-infrared fluorescence Western detection. Fluorescent dyes fall into a couple of different categories, and I'll just briefly introduce this. So there are small molecule organic dyes, which are more commonly used. So these dyes are small molecules, and their color, or their fluorescence, excitation, and emission, is determined by the chemical structure of the dye. Because of their small size, they can be conjugated to antibodies, other proteins, or a variety of other biological molecules for different applications. And these dyes are fairly cost-effective. Another class of dyes is nanocrystals, or quantum dots. So these are actually these are crystals, and the fluorescence properties of the crystal is determined by the size of the crystal. That allows fine-tuning of a wide variety of excitation-emission combinations. These tend to have somewhat more limited applications than the small molecule organic dyes because of their toxicity and their size. And in terms of fluorescent Western blotting, probably the main drawback would be that these are much more costly than the small molecule dyes. So the dyes I'll be describing today, Biodium CF dyes, are small molecule organic dyes. Biodium specializes in fluorescent dyes and other reagents, and so CF dyes are our next generation line of fluorescent dyes, specifically designed for labeling proteins, nucleic acids, and other biological molecules. CF dyes were engineered to be superior to other commercially available fluorescent dyes, for example, Alexafluor dyes, Psi dyes, or Dye-Lite dyes, by having brighter fluorescence, better photostability, and higher water solubility. Right now, CF dyes include about 20 different dye colors, ranging from the visible spectrum, so blue, green, and red dyes, to the far red and near infrared spectra. These dyes are invisible to the human eye, but can be imaged with CCD cameras and other imaging setups. So today, with regard to Western blotting, I'll be focusing on the near infrared CF dyes. These dyes are named, incidentally, for their excitation wavelengths. So a CF680 would be excited by a 680 nanometer excitation source on a 750 by 750 nanometer excitation source. Near-infrared dyes have specialized applications due to the low autofluorescence in this region in biological samples. So as I mentioned, highly sensitive quantitation in Western blotting. Another specialized application for these dyes, because they have a very long wavelength emission that actually can penetrate through several centimeters of tissue, these dyes can be used for in vivo imaging in living animals. This is an example of imaging a fluorescent dye signal in a mouse using one of UVP's in vivo imaging systems. So near-infrared CF dyes were designed to have some advantages over other near-infrared dyes that are currently on the market, specifically in the dye chemical structure. So with modifications to make the dyes more water soluble. So the near-infrared dyes in general tend to be on the larger end of organic molecules. They are very hydrophobic and have rigid structures. And this leads to poor water solubility and a tendency for the dyes to aggregate. And when the dyes aggregate, their fluorescence actually becomes quenched. So the way that dye chemists have tried to solve this problem is by typically by introducing several charged groups to the dye structure. In this case, for Alexafluor 790, you see these multiple charged sulfonate groups have been added to the dye. And while this does greatly improve the water solubility of the dye, it introduces another problem, which is typically when an antibody is labeled with fluorescent dyes, one antibody molecule will have several dye molecules conjugated to it. And with a large number of negative charges being carried by each dye, that can actually affect the isoelectric point of the antibody and make it sticky and increase nonspecific binding of the antibody to, for example, the Western blotting membrane. The innovation in the near-infrared CF dyes is the combination of using charged groups as well as non-charged hydrophilic groups such as PEG groups in order to increase the water solubility of the dye while minimizing the loading of the dye with negative charge. And an example of the effect of this is shown here. Here are two Western blots that were probed. So this is a helicell lysate, two different dilutions of helicell lysate that were probed with an antitubulin antibody, and the dyes, the blots were treated identically, except that the blot on the left was detected with a secondary antibody conjugated to Alexafluor 790, the highly negatively charged dye from the last slide, whereas the blot on the right was detected with a secondary antibody, the same secondary antibody conjugated to CF790. And what you can see here is that with the Alexafluor dye, you see a lot of nonspecific bands showing up, and most likely due to all of those negative charges making the secondary antibody sticky and causing it to bind to nonspecific targets on the blot. Whereas with its improved water solubility but minimal charge, you see a much cleaner signal with the CF790 secondary antibody. The improved water solubility of near-infrared dyes also enhances their fluorescence. And this is because, as I mentioned before, when the dyes aggregate, their fluorescence is quenched. these dye aggregates tend to show up here in the absorption spectra of the dye. So here's a comparison, 3 different absorbance spectra for these near-infrared dyes that absorb at 750 nanometers. And the dye aggregates tend to show up in the spectrum here as a shoulder peak on the absorbance spectra. And when the dyes are aggregated like this, they actually don't contribute to the overall fluorescence of the dye. So here's a comparison of the aggregation of size 7, Alexafor 750, and CF 750 showing much less aggregation in the CF dye due to its improved water solubility. And this translates into brighter antibody conjugates, and this is shown here in this graph. It's called a conjugation plot, and it's how we compare the brightness of different antibody conjugates. What's graphed is the fluorescence of an antibody conjugates that are conjugated to different numbers of dyes per each antibody, or different degrees of labeling. So for example, here we're comparing antibody conjugates with 2 dyes, 4 dyes, or 6 dyes per antibody molecule. What you typically see in a conjugation plot with any fluorescent dye is when you add more fluorescent molecules, dye molecules to the antibody, you see an increase in the conjugate brightness. And then as the dyes get close together and start to quench each other's fluorescence, you see that fluorescence will drop off as you increase adding more dyes per antibody molecule. Because CF dyes have less aggregation and quenching, you can add more dye molecules per antibody and get a brighter antibody conjugate compared to these other dyes, the Alexafor dye and the Psi dye, where adding more dye molecules per antibody really doesn't increase your fluorescence because of the quenching. And this is just another example showing a comparison of our CF770, which is a popular dye for Western blot detection, with dye-light 800 and IR dye 800 antibody conjugates, showing the brighter fluorescence of the CF antibody conjugate. So here's an example of a multiplex Western blot comparing CF dyes with, in this case, the IR dyes. And so here we have different dilutions of HeLa-cell lysate were transferred to the membrane. The highest concentration at 2 micrograms per lane down to 0.125 micrograms per lane. And here tubulin was detected using with a mouse antibody and either IR dye 680 LT or CF 680. And then in green, COX-4 detected with a rabbit antibody raised in rabbit and an anti-rabbit secondary antibody conjugated to either IR dye 800 CW or CF 770. And when the fluorescence of these bands is quantitated, what we see is that the CF dye, the bands detected with CF dyes are 1.5 to 2 times brighter than those on the IR dye blot. Here's just another example of a multiplex application, which is detecting a post-translational modification. These are lysates from cells that were treated with a MAP kinase inhibitor or activator, and detecting the phosphorylation of the protein ERK, the MAP kinase, the kinase ERK. So what you see here is detected with CF680 is the phosphorylated protein, which is present specifically in the cells that were treated with the activator, and then simultaneous detection of the total levels of the protein ERK, phosphorylated and non-phosphorylated, showing that the loading control, basically that the same amount of protein was loaded in each of the wells. There are some special considerations when making the transition from a chemiluminescence detection to fluorescence detection. The main one being multiplexing considerations. And so that is, in order to detect 2 targets simultaneously, you have to find antibodies that are raised in different species, allowing you to detect simultaneously with different secondary antibodies. And in addition, when doing multiplex detection, it's advisable to use secondary antibodies that have been highly cross-absorbed for multiple labeling. So these are secondary antibodies that are highly specific for one species and have minimal reactivity with antibodies raised in other species. While biological samples have very low fluorescence in the near-infrared region, actually some commonly used Western blotting reagents do have high fluorescence. And so loading dyes, in particular blue loading dyes that are used when loading polyacrylamide gels, can cause significant interference. And so it's recommended to use a dye that doesn't have infrared fluorescence as your loading dye, orange G being a common one. Membranes also, so nitrocellulose and PVDF membranes used for Western blotting can have some fluorescence background. This tends to be more of an issue for the PVDF membranes, and so it's important to choose a membrane that is specifically designated for fluorescence applications. As Sean mentioned, there are a number of proprietary blocking buffers that can be used because this is a highly sensitive method. Keeping background low is important, and so finding the right blocking buffer can be something that needs to be optimized. In general, when handling fluorescent dyes, it's important to protect them from ambient light during the experiment, and then during imaging. However, CF dyes have really excellent photostability, and so actually in practical terms, we've not found photo bleaching to be a really significant problem. We can image the same blot over and over without really seeing any significant fading of the fluorescence signal. So to summarize, the advantages of fluorescence detection and CF dyes. So fluorescence detection allows us multicolor detection, a higher sensitivity and broader dynamic range than chemiluminescence. CF dyes have advantages in higher fluorescence compared to other near-infrared dyes, and they're highly water-soluble for improved antibody specificity. The fluorescent signal is highly stable, so as I mentioned, we've imaged the fluorescent signal on blots, even years after the experiment's performed, without any really appreciable loss of signal. So Biodium offers a wide variety of CF dye labeled secondary antibodies as well as anti-tag antibodies, and we do offer highly cross-absorbed secondary antibodies for multiple labeling for Western blotting applications. And we also offer reactive dyes and antibody labeling kits.
Well, thank you very much, Dr. Roberts, for your in-depth presentation of Biodium's latest CF dyes, especially including information about the advantages and special considerations of working with such materials. It's very exciting technology. To wrap up our presentation today, I'd like to turn the microphone back over to Dr. Gallagher to briefly discuss UVP and its Western blot imaging solution, the BioSpectrum imaging system. Dr. Gallagher?
Oh, thank you. Our BioSpectrum is an ideal setup imaging system for doing both chemiluminescence and NIR fluorescent blotting. The available configurations come with high sensitivity cameras that are particularly sensitive out to the NIR, but have quantum efficiencies in the visible that reach up to 80 and 90%. We have a fully automated light-tight darkroom, 5-position emission filter wheel. For chemiluminescence, one of those positions is left open, while the others can be tailored to a range of wavelengths needed for the fluorescence detection being undertaken. I mentioned the deeply cooled CCD camera. We have a range of cooled CCD cameras, but specifically the camera designed for NIR imaging is also ideal for chemiluminescence. So you'll find that the very same features of sensitivity for fluorescence imaging are also perfect for doing chemiluminescence capture. We have zoom and fixed lens options. The fixed lenses tend to cover larger CCDs and come in f numbers or apertures down to 0.95, making them ideal for chemiluminescence and fluorescence detection. And in addition to the full automation of the software, the software that we have that controls the cabinet functions, we have templates which allow you to pre-program specific experiments, specific filters, so that day in and day out you can use presets that will take you all the way to final image in the same way for enhanced repeatability. The cabinet is designed for routine laboratory analysis, which includes UV excitation of gels, as well as visible light work, and as we've been mentioning, IR detection. For fluorescence blotting, the BioLite multispectral light source is ideal. It not only uses a high-intensity xenon light source, which gives a broad spectrum of illumination from the long-wave UV up to the NIR beyond 900 nanometers. It also produces a very intense light, which makes the detection of fluorescent tags straightforward with relatively short exposures. It uses fiber optic light channeling to illuminate the membrane evenly, and is software controlled for automation via templates for reproducible experiments from day to day. One of the many advantages of the BioLite is that you have 8 excitation filter positions, meaning you can leave one of them empty for white light exposure and the other 7 for a range of excitation wavelengths to fit everything from visible up to near-IR dyes. And in fact, these filters are interchangeable and can be customized, so really the full range of available dyes to the life sciences can be imaged with the BioLite. One of the advantages of the xenon light source is that the light produces a range from the long wave UV all the way up and beyond 900 nanometers. So by picking the appropriate filter, we can hit really any wavelength of interest for fluorescence detection. With an LED, for example, you would have to use different LEDs for every wavelength of interest. And lasers, of course, would have to be switched out, making it really not practical. Xenon arc lamps also are fast. They're area imagers when combined with CCD imaging. And again, comparing that to a laser that scans, takes a long time. It's fairly slow. And the cost comparison, again, between lasers and our xenon arc lamp, lasers quite expensive, particularly when including multiple lasers. So overall, the xenon system is ideal for doing all types of fluorescent blot imaging. So this leads to a simplified workflow for near-IR fluorescent westerns. By using the BioLite multispectral light source and the appropriate filters, you can excite and detect a wide range of biodium CF dyes with a particular excellence in the near-IR. We have a range of standard and custom filtration available with our excitation filters. And again, customized filters are relatively easy for us to obtain, and frequently we can have them in a matter of weeks, and specialized, highly specialized custom filters may take a little longer. With our sensitive and deeply cooled CCD cameras, we can readily go out into the near-IR with very high quantum efficiency, and the same cameras can be used also for routine visible wavelength imaging.
Thank you very much, Dr. Gallagher, for your overview of the biospectrum imaging system. And I want to thank our audience as well for submitting a variety of questions that we have to select from here moving forward. I'd like to encourage you to continue submitting questions. If we aren't able to get to yours, we will reach out to you on an individual basis after the webinar is over with. But for now, let's begin with a few of the questions that we've had coming in. With that, our first question is for Dr. Roberts. What are some of the major applications for CF dyes?
Sure. So CF dyes, basically, as I mentioned, we have colors spanning the visible to near-infrared spectrum. And so they're typically used for fluorescence microscopy, immunofluorescence detection, flow cytometry. And then the near-infrared dyes have the specialized applications in vivo imaging and Western blotting, but also are used in flow cytometry to add additional channels for multicolor detection.
Great. Thank you, Dr. Roberts. Our next question up is for Dr. Gallagher. How do I determine if all of my protein transferred out of the electrophoresis gel onto the membrane, and what is the most efficient way to perform protein blotting?
Yeah, that's a great question. Frequently, researchers don't realize that they leave protein behind in their gel during blotting techniques, and it's important to get a complete transfer in order to get quantitative data. So once the protein blotting is complete, remove the gel from the membrane carefully, and then you can stain the gel to visualize to see if there's any remaining protein in the gel. Typically with shorter blotting times in using devices sometimes called semi-dry blotters, the high molecular weight proteins are transferred less efficiently, and in fact they do tend to stay in the gel. Tank blotting systems, on the other hand, you can go much longer, and they have more buffer in them, and they are more efficient at transferring high molecular weight proteins onto the blot. But again, staining the gel is a great way to determine what you've left behind and whether it's important to you.
Great, thank you for that. Our next question is again for Dr. Roberts. How and why are CF dyes so stable, especially using Western applications?
CF dyes, because they're small organic molecules, they just have they're intrinsically highly stable chemicals, and so once the blot, with the dyes localized on it is dried, there really isn't any kind of degradation that takes place. So as long as the blots are stored protected from light, just in a drawer, for example, or wrapped in foil, the dyes continue to be highly fluorescent for extended periods of time. And in addition, one of the important features of CF dyes that the dye chemists here at Biodium engineered the dyes to have is good photostability. And so what that refers to is the stability of the dye fluorescence while it's being exposed to high intensity excitation light. And so the dye structures are optimized to have very good photostability as well. So you can image the same blot again and again without losing your signal.
Well, that's great. Thank you. Thank you, Dr. Roberts. I think we have time for one more question, and this will be for Dr. Gallagher. How do I determine if my protein is binding to the membrane?
Yeah, sure. That's and the other half of the question of what's left in the gel is to determine if the membrane has bound all the protein of interest. And particularly with nitrocellulose, if it is not stored properly, it can be stored out in the open in high humidity environments. It can lose its capacity to bind proteins. And so during the blotting process, the proteins don't stick as well. And there are a number of ways to stain the membrane to see in a qualitative sense whether the proteins have bound to the membrane or whether some have leaked through. These include stains such as Kamasi blue and India ink are ideal for this, and these techniques allow you to stain the membrane and look at the front surface to see if the protein is binding, and then also look on the back surface of the membrane to see if some protein has blown through. And why that's important is if you're trying to quantitate how much protein is in your sample of a particular protein species that you're interested in, if it's leaking away from the membrane, then quantitation is very difficult. You want that membrane to capture everything.
Well, great. Well, thank you both, Dr. Gallagher and Dr. Roberts. And again, thank you to our audience for participating by submitting some good questions. So unfortunately, we've run out of time, but for any of your questions, again, that we weren't able to answer now, we'll definitely reach out and contact you shortly on an individual basis to answer those questions and any other follow-up questions you may have. In addition, if you feel like contacting us, you can reach out to us using the contact information you see on your screen, either to UVP or to Biodium, using email address, phone numbers, or the website shown. Thank you again to both of our presenters, Dr. Lori Roberts and Dr. Sean Gallagher. And thank you to everyone in our audience for your time and participation. You'll be receiving an email shortly letting you know how to access today's event online so you can see any material you may have missed or if you'd simply like to review again. On behalf of our presenters, Dr. Lori Roberts and Dr. Sean Gallagher, as well as the entire teams here at UVP and Biodium, my name is Mike Capps, and I wish you a great day. Thank you.